Assalamualaikum warahmatullahi wabarakatuh. So in this opportunity, I want to presentation about scientific articles. My name is Ramadalia, NPM H1A020032. I'm a medical student from the University of Bengkulu. So the title of article is the MPVG VLPG projection neurons contribute to pain threshold maintenance and anxiety behavior. There are 5 sections that I want to explain. The first section is the background of this article, and then the second is the purpose of this article, and the third is the methods of this article, and the fourth is the result of this article, and the fifth is conclusion. And so the first background. There are 4 points that I want to explain. The first is physical symptoms in a patient depression or anxiety disorder. So there are often physical symptoms in a patient depression or anxiety disorder, like pain. It is often forgotten by a doctor and psychiatrist. Even a few of the primary servants for typical anxiety and depression disorder are usually unnoticed, and then often found in some sort of patients to treat symptoms, often not as a pervasive or as inappropriate. For that, this requires research on neural pathways that may be involved in early stage of pain threshold maintenance and anxiety behavior. And one of the neural pathways to be studied is the DMPVC. The dorsal medial prefrontal cortex has been recognized as a key cortical area for nociceptive modulation, and however, the underlying neural pathway and the function of specific cell types remain largely unclear. So, the purpose of this article is to show that lesions in the DMPVC indicate an agesic and anxious stage. So the point is, we want to prove that the MPVC-VLPEG projection neurons can contribute to pain threshold maintenance and anxiety behavior. And then the third is the methods of this article. There are 4 general methods. First is experimental animal. So experimental animal, that is visual to iris cream mice. So we use mice, and then there are 8 tests that we have to do. The first is the CPNL model. This PNL model was established to observe pain and anxiety. And the second is measurement of sensitivity to mechanical stimuli. And then OF and APM test. For APM test was made of a wide basic class apparatus, and next theoretics KA, tracer and virus injection, and then fiber optic virus or cannula implementation, and next behavioral tests using optogenetic, semogenetic, and pharmacological approaches, and immunohistochemical and immunofluorescence histochemical staining, and the last histochemistry combined with retrograde track tracing. And the second method is electrophysiology. So in this method, the first is coronal brain cells containing the MPVC were prepared. So transfer slices were cut on a vibrating microtome at 4 Celsius. The slices were transferred into a submerged chamber containing artificial cerebrospinal fluid containing reagents like sodium chloride, and then the recordings were made in voltaic clamp mode using the Exxon 700B PC clamp software version 10.02 was used to acquire and analyze the data, and the third is Western boat analysis. So the first, the DMPPC was collected and homogenesis, and after this, all reactions were detected by the ACL detection, and then we can get the densities of protein boats were analyzed using lab work software. And the last, statistic. So statistic statistic values are reported as the mean more or less SAM. And the first statistical analysis were performed using graph Prim's version 6.08 graph software, and we can get a p-value of 0.05 or less was considered statistically significant. And the result is the first bilateralization in the DMPC worsen CPL-induced mechanical nociception and anxiety-like behavior. And two, projection from the DMPC to the vigilate to containing neurons in the VLPG. And three, like microscope observation of the DMPC VLG RVM neural pathway. So the result indicate that CVNL triggered long-term GBAR mediated inhibition in the DMPPC VLPG projection neurons, and the fourth, neuronal activity recorded in the MPPC VLPG projection neuron in CPNL and some trait mice. And then five is optogenetic activation of the DMPPC PAG neural pathway antonet 7L induced mechanical hypergeusia and anxiety-like behavior. And simultaneous activation of inhibitory neurons in the MPPC worsen CPNL induced mechanical hypergeusia and anxiety-like behavior. And then the seven is optogenetic activation of inhibitory neurons in the DMPPC reverse activation of the MPPC or LPG pathway induced analgesic and antacid behavior. And the last is suppression of GBR and MGLUR1 in the DMPPG reverse CPNL induced mechanical hypergeusia and anxiety behavior. The result indicate that inhibition of GBRS or MGLUR1 may be a usable approach to rescue hypergeusia and anxiety-like behavior under chronic pain condition. And last is conclusion. The conclusion of this article is our study suggests that the MPPC neural pathway may participate in the maintenance of pain, their thoughts and anxiety behavior under normal conditions, while silencing or suppressing the MPPC or PAG pathway may be involved in the initial stage and maintenance of chronic pain and the emergency of anxiety-like behavior. So I think that's all about my presentation. Thank you for the attention. Wassalamualaikum warahmatullahi wabarakatuh.